Good afternoon, everyone. Welcome to our next seminar in the series of distinguished lecture series on genomics of health and well-being. Our speaker today is Professor Arnab from PGIMER, and may I please request Professor Arnab to kindly introduce our speaker for today.
Thank you, Alpana. So this is a series that was conceived by our founder director, Professor Partha Majumdar, and over the years, and we had been conducting it quite well. In between, we had a pandemic caused forced break, and then we have started this year to pick it up, and I think we have already had a series of talks, and we hope to continue these. So combination of online and hybrid events on this series. So today we have Professor Arnab Pal, who is the professor in PGI biochemistry in PGI Chandigarh. So there's a connect, there's a collaboration between NIBMG and PGI. So this is one of the hallmarks of NIBMG, that it coordinates and collaborates with this prominent tertiary care and research hospitals and medical centers all across the country. So just to give a brief synopsis of Professor Pal's career, he actually did his MBBS from Archicon Medical College, Kolkata, and went on to do his MD and then PhD from All India Institute of Medical Sciences in New Delhi. And subsequent to that, he joined as assistant professor in the GRIMS in Shillong. For about 3 years. After that, he moved over to PGI Chandigarh as assistant professor, and from there he had a meteoric rise to recently. I think I'd remember congratulating him when he became professor. So he has a whole lot of awards. I mean, including the AACR Scholar in Training Award, Rapid Grant for Young Investigators of DPT, member of National Academy of Medical Sciences, and recently, some of us had the good opportunity to congratulate him, and you might have seen it on Facebook that he has been nominated for Mrs. and Dr. G.P. Talawar Oration by Association of Clinical Chemists of India. So that is a very, very distinguished achievement, and we congratulate him as an IBMG in a probe to you again, Arnab, so Professor Paul is a well-wisher and a friend and a collaborator of NIBMG. I will not go into his details, like he has like more than 80 publications already, a book chapter, 12 grants. He's a member of various important bodies in the country, like Association of Clinical Biochemists of India, Indian Association for Cancer Research. He's an executive committee member. Dr. Sandeep Singh from our institute is also there. And then a member of Association of Medical Biochemists of India and various other organizations. He had received many awards, as I said, and he's guided 24 students for various degrees like PhD, MD, MS, DM, MCH. So Professor Paul and I we actually collaborate on oral cancer, some specific studies on oral cancer. So I had the good fortune to interact with him both as a collaborator and as a close friend. And what I have found about him is that he is very knowledgeable and he is very enthusiastic, and most importantly, he is very helpful. So we look forward to his involvement and engagement with NIBMG more in the years to come. And beyond that, I think I will close now and not stand between us and his talk. Professor Paul.
Thank you very much, Dr. Maitra, for such a elaborate introduction. I'm humbled to be here and thankful to the whole NIBMG family to make me present here today. As Professor Maitra has told, I am a doctor by degree and shifted to research. So I'm a very small kid in the research field, biomedical research field. Here today, I just want to share with my journey towards some of cancer research. So today, the title of my talk is Biomarker Discovery for Oral Squamous Cell Carcinoma, Candidate Gene versus Omics Approach. My laboratory is a very new one, only 7-8 years old. We work in 3 fields of cancer and all are different kinds of biomarker discovery. So today I will be talking about proteomic biomarker discovery. So just to brush up, what is a biomarker? As per National Cancer Institute, NIH Bethesda, it is defined as a biological molecule found in blood or any other body fluid or maybe in tissues. And it is a sign of normal or abnormal process or a disease condition or a reaction to a disease condition. And these are used for identifying how body is responding to the treatment or a disease condition or so on and so forth. Now, the biomarker can be of cancer related or maybe non-cancer related also. So as a cancer researcher, we are more interested to see the tumor markers. Again, another sister concern, National Cancer Institute, NIH, they have defined the specific definition of cancer marker substances produced by cancer or by normal cells of the body in response to cancer or certain benign conditions also. The red part is important. They are specifically produced by pre-malignant or malignant tissues. They has to be produced in a detectable level by all the patients in a specific malignancy, expressed in an organ-specific manner, present in different body fluids or tissue which are easily accessible, and the levels of these markers should relate quantitatively to the tumor volume or biological behavior or maybe in the disease progression. And finally, I'm a laboratory physician. The last point is very important for me. For a patient treatment or diagnostic viewpoint, it should be easily measurable by standardized, reproducible, validated quantitative assay. Now, as we know that definition, it can be site specific. It has to be measurable, detectable in different body fluid as well as in a quantitatively expressed. Now, if I talk about the different types of tumor marker, as I was told, it can be a diagnostic marker for diagnosis of the treatment. It can be a predictive marker, which can predict the efficacy of a treatment or even a drug. It can be a prognostic marker, how disease is behaving, how it is going from stage 1 to stage 2 or stage 3, stage 4 metastasis and all those things. And it can be for staging. So all kinds of markers are there, and my laboratory is looking into not only diagnostic but also predictive and prognostic markers also. But today we are morely concerned about the diagnostic marker. Now coming to the some very technical but boring but important criteria, that when we define a marker, we have to follow some reporting quality that's known as BRISC quality, biospecimen reporting for improved study quality. These are very basic for any biomarker definition. So the elements of BRISCs are prioritized into 3 tiers. I'm not going to detail. There are so many sub details are there. It can be items recommended to report, that includes information such as which organ it has come from or anatomical site, which biospecimen, how they are collected, how they have processed and how they have stored. This is very important for any kind of biomarker. For example, it has been taken from serum or urine or saliva or whatever is the thing. Now, second criteria is slightly lesser in importance, that items beneficial to report is slightly less crucial in scientific contribution, such as the time of biospecimen acquisition or acquisition or stabilization, that when it has been collected 2 days back, 7 days back, something like that. And there are some additional items to be reporting. That is, what are the conditions useful to know for concerning the biospecimen, like environmental factors. Now, when we report a biomarker, there is a clear-cut guideline. Otherwise, neither it can be published, nor it can be used as a clinical purposes. This criteria is known as remark criteria, and this has been in practice till 2005 after a Nature Clinical Practice Oncology purpose. Remark stands for reporting recommendation for tumor marker for prognostic studies. The goal of this remark criteria, quote unquote, the goal is to encourage transparent and complete reporting so that the relevant information will be available to others to help them judge the usefulness of the data and understand the context in which the conclusions apply. Now, coming to the problem of our country, where do we start? As we know, there are many distinguished faculty in front of me who work on the oral cancers. This is a typical problem of our country. The Western population, they are not much interested in working in the oral cancer because it is not their problem, it is our problem. So oral cancer refers to any cancerous growth located in the oral cavity, and 90% of these growths are related to histological types of squamous cell carcinoma, or commonly known as oral squamous cell carcinoma. They arise from lips, gums, tongue, alveolar, mandible, and all those, so on and so forth. This is sixth most common cancer in the world, but 62% of this cancer coming from the countries like ours. And in India, oral cancer ranked third as per the 2020 Globocan, just after the cervical and lung cancer. Now, as we all know, those who are working in the oral cancer, they know very details, and I'm not going to detail that tobacco smoking, be it a smoke or be it a smokeless tobacco like betel quid, oral snuff, and all those things like gutka, khaini, which we know as common terms, they are the major risk factor for oral cancer. Erica nut and alcohol are also additive risk factor. There are some minor risk factors like genetic factors, HPV infection, and chronic trauma, which are also associated with oral cancer. If we talk about the diagnosis of oral cancer, mainly the clinical examination supplemented by fine needle aspiration biopsy or fine needle aspiration cytology along with radiological examination like CT or MRI are the diagnostic tools what we use for oral cancer. As per current practice, tissue biopsy followed by histopathological evaluation of the tissue specimen is the gold standard for diagnosis of oral cancer. But if I talk about the problem statement of our country, 2/3 of the cases of oral cancer come to us at the later stage. And as a result, there is very high mortality and morbidity. So we were thinking that evaluation of a tumor marker for OSCC for early diagnosis, will it be successful or not? Now, regarding the treatment of oral cancers, surgical therapy is the mainstay of therapy throughout the world. Adjuvant therapies like radiotherapy or chemotherapy may be used in different combination depending on the different stages. In our country, our setup, wide local excision along with neck dissection and 66 gray of alternate fraction radiotherapy is a standard curative therapy for oral cancer patient. For the last 2 decades, there is a tremendous advancement in the treatment procedure for all cancers, including oral cancer. However, even after that, in our country, 50 to 60% of the cases does not go for a 5-year survival rate. So it's a very abysmal, very bad prognosis of oral cancer. This is bothering us. So when I joined in PGI 2011, Professor Sushmita Ghoshal, who is the currently professor and head in the radiotherapy department, who is looking into the oral cancer department, one day catch hold of me in the faculty cafeteria and tell me that you have come, you have done some research, so kindly help me to solve this problem. The problem, what we identified in that coffee house, that there is no tumor marker till date. Actually, if we get a tumor marker, we can identify even the cancer in a very early stage. There is a term known as catch them young. Here we can catch them early. If we can get those patients at the early stage, then half of our problem is solved. Because in our data, we found that point of time itself that those who have come into stage 1 and 2, they have almost 80% 5-year survival rate with the same treatment. On the other hand, those who have come into stage 3, they have only 40 to 50%. So catch them early was the motto. Now, how to catch them early? So we identified in that cafeteria that biomarker identification may be an important part. Now, again, going back to the theory, biomarker identification approach, we read so many books and all these things. So we found that there can be 2 approaches. One is the candidate biomolecule approach, by which candidate biomolecule is searched from the literature by different publications and identified some of these molecules and validate them in the patient along with their control group. There is a standard procedure. Along with that, there is a newer recent advances, that is the OMICS approach, which can be genomics, transcriptomics, proteomics, metabolomics. Your institute is a landmark institute in India in omics, so I don't have to go into the details. You know better than me in the omics approach. And from that omics approach, using different bioinformatic tools, we identify some disabulated molecules and validate this. So initially, when we are starting in 2011 or 12, we didn't have the approach to this omic approach even at our institute. So we started with the candidate biomolecule approach. We have done a thorough literature search. Okay, what are the genes and all those things can be taken up, or protein. So we identified a candidate protein and validated it in different stages and different types of tumors. The protein was identified by us as a candidate gene as a crypto1 for a diagnostic biomarker. It's a EGF-CFC family protein, and the molecular weight is between 24 to 36 kilodalton, depending on the amount of post-translational modification. This is encoded by a teratoma-derived growth factor gene, and peculiarly, this is an oncofetal protein. This protein expressed during the fetal period and later on during oncological, in some cancers, this also expressed. This was found to be in literature for cell proliferation, motility, survival, epithelial mesenchymal, all kinds of cancer-related diseases, and found to be expressed in a group of cancers: nasopharyngeal, breast, cervical, gastric, uveal, or whole body cancers. Even that point of time in 2011, we found an article where Hun et al., although he is from a Chinese group, but the article we found that it was substantially good. So we found that article that was new that point of time, that crypto1 was overexpressed in the tissues of oral squamous cell carcinoma. And we got intrigued. Can this be a biomarker for us? So we planned our first research project that let us measure serum crypto1 by ELISA. Unfortunately, we found a kit is also available by R&D Systems, which is a decently okay biomedical company. And fortunately, we got that in our patients, there is a significant raise in the crypto1 level in our patients. And it is in the both in the early stage of the patient as well as in the late stage of the patient, which is a first hurdle we have crossed. That is, it can be a biomarker. First thing what we have understood. Next thing, we followed up the patient after the standard therapy, what I have told, that surgery followed by 66 gray fraction curative intent surgery. Yeah, we included only curative intent patients only. And we found that there is significant fall of crypto1 after surgery in the early stage patient as well as in the late stage patients. That means it again fulfills another criteria to be another biomarker. We got interested that there are two groups of patients also, which one group are cured after therapy, another group there are residual disease after therapy. What is the difference between them? Fortunately, we found that in the group where after therapy there is no clinical or radiological evidence of disease, the crypto1 has fallen significantly. But the other group where there are residual disease is there, the serum crypto1 has not fallen significantly. Although there is a low, but it's not significantly fallen down. So the three criteria of a biomarker, what we have done told as the NCI criteria, has been fulfilled by crypto1. And then we analyze the diagnostic ability of this crypto1. We found that it has an area under the curve, which has a significant sensitivity, specificity, and 0.8 area under the curve, and sensitivity, specificity at 74 and 78% with a cutoff value of 202.5 picogram per ml. So till now, we found that it can be a very potential biomarker for diagnosis of squamous cell carcinoma. So we want to check the Chinese group's data again. Is it true for our purpose also that what they have got in the tissue level, are we getting it? And fortunately, we also got it that OSCC tissue has significantly high level of tryptophan level. So we concluded that increased serum tryptophan level, which go down upon treatment and correlated with the early disease condition, suggests that this can be proved to be a potential prognostic as well as diagnostic marker of OSCC. We did it, publish it, but we are not happy with this. Why? Because to put it as a tumor marker like prostate specific antigen or for ovarian cancer CA, that is a since specificity is very important. Means only for this disease this has to be increased. But in our case, the literature itself says it increases in multiple cancers like nasopharyngeal cancer, cervical cancer, breast cancer, so all plethora of cancers. So it lacks specificity. Although it has a very good sensitivity as a diagnostic marker, but it lacks specificity, where we are not happy with this marker. So then by this time, 2 years has gone, we got some grants from DBT. The first part was done from a grant from DBT, and after with this grant, we got another grant from SERB. So we got some more funding to explore the other aspect of the biomarker discovery, the OMICS approach. What is the OMICS approach? It can be genomics. You can teach me this, no problem at all, because this is your niche area. Genomics, transcriptomics, metabolomics, and proteomics. So these are all omics, different kinds of omic approach for biomarker discovery. Now we have to decide which omics approach we will take. I didn't have collaboration that point of time with the NIBMG. Had it been there, I could have gone for maybe genomics or maybe transcriptomics. The omics, genomics based discovery you are already doing in this institute in oro gingival cancer itself. So in those cases, whatever we had, we targeted to proteomic approach. This I'm not going into detail because you know, okay, what are the genomics, next-gen sequencing, exome sequencing, DNA sequencing, and comparative genomic hybridization. This we know better than me. There is transcriptomic approach where you can have microarray RNA sequencing, non-coding RNA sequencing, all these things you do routinely. Coming to the proteomic approach, there are 2D, DGI, cell of MS, gel LCMS, and ITRAC. These are the different parts where we can get it. And metabolomic approach, GCMS, LCMS, MS, NMR, we can use it for different purposes. Now we have to decide what we have to do. Two things we have to decide: which omics approach we will do and on which tissue or biological fluid we have to do. Again, we sat on a cup of coffee and we found out, let us do something which people has done slightly less. It is not that less. So we identified that let's do it on saliva. Salivary proteomics. Don't do it on the crypto one part is done on the serum, but we'd like to do it on a salivary component. Saliva. Why? For oral cancer, saliva is a contact fluid. The oral cavity is always bathed with a saliva. So whatever cells or cellular component or proteins or whatever debris or metabolome that directly come into saliva to the patients. Okay, it is a, if I ask a patient that please donate me 5 ml of blood as a recruitant of this study, we want to do research on you, they may not give it. But if I ask him to just spit 5 ml of saliva on this, they may be easily giving it. So it is a practically, it is a easier one. And second point is that we wanted that we should go to the outreach. At the end, we should go to the do something which we go to the screening to the remotest corner of our country, where maybe the doing in the blood will be a problem. So by taking in all these things, all this consideration into our point, we have identified, let us do it on saliva. Then we again searched the literature and found out there are articles where saliva has been used as a diagnostic fluid for proteomics as well as transcriptomics and other purposes also. We drew a work plan for salivary proteomics. What we did, we collected saliva sample for oral squamous cell carcinoma patient along with healthy volunteers. We process the saliva. For the saliva collection, there are two methods. One is stimulated saliva, another is baseline saliva. We collected normal saliva. In case of stimulated saliva, we put a drop of acetic acid or some nimbu lemon drop in the mouth, and so that we stimulate the salivary production and do it. But our idea was that let us keep a baseline saliva. We just clean the mouth, ask the patient to sit there for half an hour, then spit it in a 50 ml centrifuge tube. So after doing this, we process the saliva, just centrifuge it and take out the supernatant and further process it. We did a TMT-based shotgun proteomic profiling of the saliva samples for quantitation of salivary proteins in OSCC case related to the healthy controls. All the cases were included those who have not been treated previously for any purposes. And the healthy controls, they are clinically and radiologically, they didn't have any kind of infection of the mouth or oral cavity. Then we have identified a group of candidate proteins from the analysis and finally try to validate them by different processes. And finally, we have tried to identify some salivary biomarkers. So let us see how we did it. So in the first phase of the discovery phase, as you also do in the discovery phase, we have taken 5 groups of the patient. As we know that smoking, chewing, smoker and chewer and no tobacco user, there can be different etiology of oral cancer. So we have taken from each and every etiology 5 samples each, and we have a healthy control group also, and then it did a TMT based identification of our dysregulated proteins. Now, after we get this method, we have done a bioinformatic analysis. Again, this bioinformatic analysis, most of you know 10 times better than me at least. Okay, the how to identify a candidate gene. So we did gene ontology by using Panther with a protein class, molecular function, pathway identification, and biological processes, along with that different protein-protein interaction by strings. And finally, we have identified the gene by different cohorts, like where the chewers only, smokers only, no leaks, and all cohorts, and identified 172 proteins which has been dysregulated between different groups with a cutoff of 2-fold cutoff. And there are 130 proteins were dysregulated either of the 4 cohorts. These are the Venn diagram where for different groups how many proteins are upregulated or downregulated. So we got a group of protein which has been upregulated. This is a fancy heat map where we can get the 71-fold, 2 to 71-fold upregulated protein in different groups, and there are similar heat map is there where they are downregulated also. Again, we are at a fix. Okay, how to do it? So we got this information and we started again literature surf. So it's a kind of omics as well as candidate gene approach. I did trying to 1 genes and trying to validate them. So similarly, we found a gene, interesting gene that is known as cornulin. Now this cornulin, when we started doing in the cornulin, we found that only 5 or 6 articles there in cornulin. So it's a relatively unexplored gene, and it has been 5-fold downregulated in our patients. So we take a risk. It's a negative biomarker, not upregulated. The downregulated things also can be a biomarker. It's a negative biomarker. And we found that on the information from literature, it has a 2 helix loop helix. That means it can bind to some calcium binding proteins, that EF hand calcium binding protein. It has a C terminus, which is a glutamine and threonine rich repeats about 60 amino acids and members of fused gene. But there is no function of cornulin has been reported in literature by that time. Only reporting 2-3 articles related to cancer is that it is discovered in esophageal stain and found to be esophagus specific in 2000 by one Chinese group, and expression of squamous epithelium like esophagus, cervix, vagina, and all those things. And functional epidermal differentiation role in cell cycle was documented but not explored in detail. Again, we started to see what happened, and we found that there is a significant downregulation of cornulin in our patients. When you go for the different differentiation, we found that, as reported in the literature, cornulin is downregulated significantly in moderately differentiated and poorly differentiated in comparison to the controlled and well differentiated. So it is also downregulated in with the differentiation. We put a survival curve and found that with a lower cornulin level, there is a significant risk of lower survival. And those patients has been followed up for almost 3 years. So with this thing, we can see that lower cornulin can be a prognostic marker, salivary cornulin level. When we look for its diagnostic property, we get a carry under the curve 0.66. Below 0.7 or 0.75, we don't consider is a very good diagnostic marker. So as a diagnostic marker, cornulin is not a very good marker, but as a prognostic marker or a marker of differentiation, it is a good marker. Similarly, we want to check that what is the status of cornulin post-therapy. We found that in case of NED group, that is no disease after the therapy, we found there is significant increase in the cornulin level after this follow-up. So it can be, again, it gives us a good prognostic marker criteria in comparison to when the residual group, there is no significant change. So with this OMICS and candidate gene mixed approach, we found that salivary cornulin level can be used as a prognostic marker for oral squamous cell carcinoma. We go on checking in the tissue level also, and we found that in case of normal squamous epithelium or a psoriasis is the hyperproliferative skin disorder where the normal squamous epithelium proliferates in a very high rate. The expression of cornulin is very high, where in case of well differentiated, it is there, but in case of poorly differentiated, the cornulin level is practically nil. And this is quite significantly when we analyze it by the ISC scoring. So we concluded that significant downregulation of cornulin levels in tumor tissue in OSCC patient in comparison to the tumor tissue, and tissue cornulin also inversely correlate with the grading of the tumors. We can get a decent idea about a prognostic marker also, but as a diagnostic marker, it does not have much significant role. We tried this approach in some more proteins also, like PSAP. PSAP is another protein where you can get area under the curve. We can get an increased level, but it is not very significant. Although p-value is significant, but the area under the curve is 0.6. I don't, as a laboratory physician, I don't take it as a very good diagnostic marker. Although there is a significant role in prognostic, but we're not happy. Similarly, we are doing on lipocalin and kallikrein, although the levels are high in the serum and saliva. In the kallikrein also in the saliva is high, but not very good. So the same question is still bothering. None of the candidate molecules is a very significant molecule to be used as for clinical practice. For paper publication, for a diagnostic, it's okay. For us, we have published the papers also significantly, I have shown, but the problem is that for a clinical purpose, when we use in a clinic or patients or something, none of this has a very high thing. So again, we have gone back to our strategy that let us do something else from our omics data. So what we tried a targeted proteomics by using parallel reaction monitoring. So as I have shown you that there are a 27-28 group of number of proteins which have been upregulated significantly from 2 to 78 folds in our salivary proteomic. Let us validate some of them together itself and find out what is happening. So in case of PRM, the strategy is that in a single mass spec run, you quantify more than one protein. So in our case, we targeted 15 proteins with 28 different peptides. So this is a very typical quantitative proteomic strategy where we use peptides which has been designed according to the PRM strategy as described by Naveen Ruinier in 2015 from outside and standardize it for a standard curve and then quantify in our patients. So these are some criteria where I'm not going into the details of them. And these are the proteins, 15 proteins we have selected and one or more than one peptides we have selected. And these peptides are heavily isotope level peptides. And these are used for a initial method development. The advantage of ELISA is that the method has been developed by someone, you are just using it. But the problem of PRM is that you have to develop it first, validate it from your own samples, and then use it for the actual samples. So in these cases, we use light peptides as well as the heavy peptides in a particular fractionation mode and do a data dependent analysis for M by Z ratio as well as retention time. And finally, we get a full MS and followed by a PRM data. And this method development is also revalidated by a reverse calibration where we mix the heavy peptide and endogenous peptide in a different concentration and from a range of 0.5 femtomole per microliter to 256 femtomole per microliter and finally draw a standard curve of a wide range. And finally, we identify a lower level of quantification and finally we by the area under the curve, we measure the concentration of endogenous peptide. We use this data by the Thermo Fisher software, what is Skyline, as well as and later on by using the analyze the data by using R. What we got? We started with 15 proteins, which has been significantly upregulated, but we could have got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, only the proteins. 3 we cannot identify in that our M by Z window. All were upregulated, but in our data, we found that only 4 are upregulated and the rest are downregulated. The data has got completely changed. 1/3 is upregulated and 2/3 is downregulated. This many times happens with discovery phase and the kind of study we have planned. We pulled the data in the preliminary sample and this happens. Now, how to dig something out of this data? What we have done? We have taken a twofold. We have sorted those proteins which are twofold upregulated in our PRM data and or twofold downregulated. So this we sorted these 6 proteins. Out of these 6 proteins, 2 are AHSG and KRT6C. These are upregulated significantly. And in our, you can see in our patients group, they are significantly upregulated both the proteins. And 4 proteins were significantly downregulated also. So in a single experiment run in a group of our patient, we got this information. But when we analyze this information for individual group, again the area under the curve is coming around 0.6, 6.8. Are we getting it something out of it? Actually, no. Similarly for KRT6C, 0.64. Similarly for kallikrein 1, 0.66. Similarly from PPIFP2, 0.66. And AZGP is slightly better, 0.72, just cross the threshold. So again, we had came back to in a position where we cannot conclude much. Now we have used some strategies. The strategies you use every day. So initially significantly dysregulated protein, we removed the non-significant one and put a simple statistical tool, univariate logistic regression. Those who have significant, then we put multivariate logistic regression and developed a model. So instead of using a single protein for a diagnostic marker, we combined their diagnostic ability together by using this strategy. What we got? We got that if we combine these 4 proteins were significant, so we got when we This 4 protein is significant from this univariate logistic regression, and we put a multivariate regression. When we use these 3 proteins, we get a area under the curve 0.82. Individually, they were below 60, except 172, 60, 66, 172. But when we cumulatively put their diagnostic ability, it has come to a decently okay diagnostic pattern. We get an area under the curve 0.82 with a positive predictive sensitivity, specificity, positive predictive value, negativity, almost everything above 75, around 75, which is a for a diagnostic biomarker, which is a decent thing. And we have used this for a different stages, nodal stages, and all those things we have used it. And in most of the cases, the significant level is quite high in our expected level. So what we have started and what we have concluded till now, we have started that individual proteins can be used as a biomarker, but not suitable for clinic. We can use multiple biomarkers for a diagnostic purpose, which is a suitable for thing. One information I would like to give it that these 3 protein AHSG, KRT6E and AZGP1, when we use them as a prognostic marker, we didn't get a significant prognostic marker. This is because maybe the patient number was low because of our logistic problem, because PRM is a significantly costly affair, okay, which cannot be done in a routine cases. So we concluded that omics approach for a new biomarker discovery is a far more better approach than a candidate gene approach. However, it is significantly costly and required a high throughput instrumentation. On the other hand, the bioinformatics tools where a physician or a laboratory physician like me may not be very acquainted with, okay, it plays a very important role in this approach, and discovery phase must be followed by a validation phase. Now, I have shared some of my experience here, but the story does not end here. According to Rabindranath Tagore, Vipulaye Prithibir Kototuku Jani. Shakespeare told the same thing. There are many more things on a heaven and earth, Horatio, thou dreamt of yourself. The story just started. The work is still going on. Now what we are doing, we are validating these 3 proteins in a large cohort of patients. And fortunately, we got some ELISA kit, and we are making some ELISA kit in our department also, our department. And one important thing, what is missing in our data, that is what is the status in oral pre-malignant disorders. There are as those who are working in the oral cancer, they know that there is a cancer and healthy, there is a gray zone. Oral pre-malignant disorder like submucous fibrosis, erythroprakia, leukoplakia, so on and so forth. So these patients can be a very important cohort which is missing in our current data. Now currently we have done the analysis of them and following them up for a longer period for time. And finally, we are trying to develop a point of care screening kit where we can use these 3 proteins for a screening purpose in a remote village also, where when a smoker comes to you, just put a spit of saliva into a card and find out that how significantly risk he or she has to develop a cancer, or he has already developed a cancer, yes or no. In our data, if we used all 3, we can successfully predict in 82% of our cases. So that is a significant amount of number. These are some publications with this proteomic based, and I acknowledge Department of Biotechnology, Department of Science and Technology, ICMR, and fellow Council of CSI for giving us grants. And the most of this LCMS work has not done in PGI. Only the patient and follow-up and the ELISA and validation part is done in PGI, but the proteomic part is done in the Institute of Bioinformatics, Bengaluru, and Enapa University, Mangalore. And finally, I must acknowledge humbly our patients and controls who are considered for the study, otherwise it could not have been done. This is our group. The most of the work has been done by this lady, Dr. Anu Jain, who is a postdoctoral fellow currently in Columbia University in New York, and Ritu has done the work on the crypto one. And these are my current students. The Rajan has done the work on cornulin, Geeta and Angshika, they are doing some triprotein peptide thing, and Smriti is doing the lipocalin thing. Angshika, you may have seen her. She was here for 4 or 5 months. And this lady is the inspiring force behind us. She is a clinician, but all the time behind all of us, okay, we have to do something for our patients. Thank you very much.
Regarding the combination biomarkers, whether only one combination or the peptides could be combined in multiple ways.
When we run the PRM, all the peptides are run in a single run. Okay, when we analyze them, okay, I'm just showing you we have analyzed in a multiple ways. I'm just showing you the positive data only.
No, that's fine. So that means multiple combination of peptides are possible to create ROC curves. So each ROC is from one combination.
What we have done, if you see it, I'm just showing it. I'm just showing it. For a single protein, most of the cases, one peptide is used, and the best peptide what we have used. What are the best M by Z ratio and the area under the curve while we standard curve, the R value of the standard curve, we have taken that data when we are showing it the value. Yes.
The last one is particularly missing.
This one?
Yeah. The one which is,
if you see this is a cumulative ROC curve of N0 cases. Means those are nodal negative cases. How significantly we are able to identify nodal positive and nodal negative cases by using these 3 ROC curves. These are the, with all cases, these are the late stages, these are the N plus and these are the N0. These are 4 ROC curves for diagnosing different types of disease condition. These clinical types of disease condition, clinical and pathological types of disease condition.
The N01 is a complex of how many peptides?
This is a 3 peptides, three proteins.
Right.
All these are for 3 proteins.
Fine. It's very nice. Now my question is, when you combine them,
Exactly. That is here work is going on. It is we have done it in a method that is PRM, which cannot be used for routine clinical purpose. Okay, that's why we want to make it easily available for clinics. So we are doing it in by ELISA method and trying to establish a point of care method where we get a yes or no kind of result with a particular cutoff.
Yes. Also, these 3 peptides, are they kind of part of the same pathway signaling pathway?
Actually, no. Actually, no. The biological role of these proteins are currently done by my current students. They are exploring into the what is the exact biology of them, because here we are taking care of only the biomarker property of them. Thank you.
Stability. Second, we do compare it with the aggressive and non-aggressive items.
First question. If I talk about the peptide stability, okay, the peptide for standardization, what we have got it from, that is outsourced and we bought it. So, okay. And according to their literature, if we put it in a correct minus 80 degrees centigrade temperature. According to their literature, it is stable for at least 1.5 years. Okay, JPT Biologicals are the supplier from Germany. And regarding the sample stability, okay, if I talk about the sample stability, we have validated the sample stability at different time frame by ELISA, not by PRM. The PRM, it is a technically challenging and we are not able to do it. But by ELISA, what we found in our salivary samples, okay, 6 months stability is perfectly fine, unless and until we do repeated freeze and thaw things. We have done it multiple samples at different time frame. We have analyzed for different proteins. Protein to protein, it varies. But if we don't freeze and thaw, there is minimal downfall in the level from 0 to 6 months. So I can simply say that 6 months stability is there when we keep it at a minus 80 degrees centigrade. And regarding the second question, the aggressive and non-aggressive oral cancer. Okay. I cannot say that the aggressive and non-aggressive property of the oral cancer, but according to TNM staging, we categorize them early and late, TNM 1 and stage 1 and 2, which are T for tumor size, N for nodal positivity, and M for metastasis plus. And so by this combination, early and late, we combined 1 and 2 for early and 3 and 4 for late. So they actually does not related to clinical aggressiveness of the tumor, because some TNM stage 1 tumor may be aggressive, while the stage 3 or 4 tumor may respond to therapy and non-aggressive also. And regarding another thing, if we want to check the aggressiveness of the tumor, we have to keep the patient untreated, which is ethically not possible. So for regarding aggressiveness of the tumor, I cannot comment at this point. The PRM validation cohort is 50 in each group, okay, which is a logistically okay sample which you can afford by that time. But currently what we are doing, that validation cohort by ELISA, what we are revalidating, that validation cohort contains approximately 200 plus samples with all different time frame follow-up levels, like 0 lab pre-treatment, 6 weeks after completion of treatment, and every 3 months samples what we are currently doing. Total tumor. Unfortunately, COVID has made our sample size reduced by almost 2/3. Two are overexpressed, one is underexpressed. We have not checked about their copy number variation, because we have not done any genomic study in our data. Only the salivary level what we have done. And if I talk about the literature thing, because our main goal was to can we use them in clinic or not? That was our main goal. So currently we are exploring their role by those who have been downregulated in our current cell lines. We are looking into their effect by those who are upregulated, but just downregulating them, and those who are downregulated, upregulating them. We are looking into the different aspects of our how they are behaving in formation of tumor, or what is their role in actually oral cancer tumor biology. Is there any genomic copy number variation? I don't have. Frankly speaking, we have not checked it at this point of time. So I'll take a note on this, and definitely we'll look into in the next level.
Comment on because this is discovery of biomarkers, the ROCs will be slightly inflated just because of the fact that we stated in the first slide, which is that patients come at a very late stage. So whenever you see essentially at the cancer for a very long period. And I would imagine that your samples would also have is
you towards the
so, but I mean, 80% is a great number. I think large scale validation or somehow, while I don't know how you do it, that's a solution to this, but you have to somehow validate it. We have had very recent diagnosis of cancers, and it would be possibly best if we can this in screening where the cancer is not evident or in every cancers.
I fully agree with you. There is no disagreement on with you, and the same thing what we are doing, that is the thing what we are trying to do, that validation in a large cohort of patients, and in this cohort we try to make them equally and find out what is the status in OPMDs also, that what it is happening. And for screening purpose, unless and until we screen those pre-malignant patients or pre-malignant subjects, I should not tell them patients, pre-malignant subjects, we cannot comment on their screening ability itself. So this part is your comments are, we are trying to take care currently.
So tumors in
All cases, because the what happened, PRM we can do in around 50 patients only, due to simply it's a cost effectiveness. Okay, so as a result we didn't go for any sub categorization, because then the power of the study will come to such a low that we cannot comment on anything. Okay, so these are all OSCC patients, but when we reanalyze it with a larger cohort of patient, I'm sure I'll be coming up with your answer that it's a chewer, smoker, or which group, what is happening, what. Irrespective of their habit.
the diagnostic.
This diagnostic. This diagnostic. Basically, there are two groups. One group is control group, who are clinically there is nothing, and another group is who has the diagnosed, histopathologically diagnosed OSCC patients. They were selected by clinical performance, and we have checked them for they don't have any OPMDs. Then their first degree relative, they don't have any history of any cancer. Yes, they are also chosen of the habits. They are also similar combination, because we have not when we take the patient, we don't screen out according to their habits. Later on, that habit thing we later on our controls are random controls. They are not pre-biased, prejudiced controls.
Just imagine that it's coming from tobacco. Okay, and just by random chance, you have got your controls would not have any habit of tobacco. So this will also show a very high ROC.
Yes,
so,
I agree.
So good. I mean, better than a random choice, a better choice is to weigh it according to the number of people who have the habit. So you know people, so you know suppose P proportion of people that are smokers. So your control also includes P proportion of people who are smokers, because that controls are easy to choose. We might have more individuals from which to choose them.
So in our case, the control is only where what we have chosen, that is the age and gender matched, because it is a very skewed gender ratio in case of oral squamous cell carcinoma. In our data, it is almost 85:15 male is to female ratio. We try to maintain that. Okay. And regarding the, I'm just sharing with you the proportion of smoker, chewer, and all these things. They are almost, there is no significant difference between the patients as well as the control groups. We have that is a retrospective analysis. Fortunately, what happened later on, we found out they are actually matched. We didn't match them intentionally, but it turned out to be a matched.
Pleasure, honor, Arnab, on behalf of NIPMT, such an excellent researcher. We long for, like, for our majority of us, we run after them. Difficult to get hold of such people. Congratulations.
Thank you very much. I must thank the whole NIBMG family, and I like the discussion. That's a very, very important. That's the most important thing what I have liked it. Like many places I give talks, but there is no discussion. But here, it is one of the best thing what I have understood. Thank you. Thank you very much. And thank you, Professor Macho, to invite me here. And thank you to listen to my boring talk. Thank you very much.